Hello everyone, welcome to BioBoost. Today we will cover another cell organelle named as endoplasmic reticulum. In the year 1945, these lace-like membranes of endoplasmic reticulum were first seen in the cytoplasm of chick embryo cells. Means it was the year when these membrane-like structures were first seen in the cytoplasm. They are membrane-bound channels seen in the form of a network of delicate strands and vesicles in the cytoplasm. Also, they are single membrane cell organelles, means these do not have any inner or outer membrane. These are single membrane cell organelles. And these form an interconnected network of tubules, vesicles and cisternae within these cells. These three are elements of ER which we will study further. So, tubules, vesicles and cisternae are the structures which form the whole ER. ER are considered as one of the components of cytoskeleton along with microtubules, microfilaments and intermediate filaments. These three are components of cytoskeleton we all know, which provides structure and flexibility both to the cell for its functioning. So ER is also one of the component of cytoskeleton. It is important, you should know this. Now, three scientists who first observed these structure was in 1945, and these were seen in the as a form of network. So Potter, Claude and Fullman, these are the three scientists who first observed these. Out of these, Potter and Fullman in year 1952, they were the two scientists who termed the endoplasmic reticulum term to these structures basically. Location wise, these are present in almost all eukaryotic cells, and these are found to be absent in mature RBCs, ova, embryonic cells and prokaryotes. Means only in eukaryotic cells these are present and absent in these three, and occupies most of the cytoplasm. Amount wise, these varies in amount from cell to cell. In spermatocytes, it is represented by a few vacuoles only. In the cells of adipose tissue, it is quite simple, having the form of a few tubules. The cells that are actively synthesizing proteins such as liver and pancreatic and fibroblast cells have abundant ER because these are synthesizing proteins actively. Now, in endoplasmic reticulum forms 30 to 60% of the total membrane in a cell. Okay. This endoplasmic reticulum is 30 to 60%, making the total membrane of the cell. Now, origin of endoplasmic reticulum. At present, manner of origin of the endoplasmic is not definitely known. But the most concrete hypothesis is that ER is budded off from the nuclear envelope, and it was discovered in 1974 by Wischniger. Means it is basically budded off from the nuclear envelope. Now, the basically two morphological types of ER is present. One is RER and one is SER. The smooth ER seems to arise from the rough means RER by detachment of ribosomes. Ribosomes are present on the surface of endoplasmic reticulum. If you remove these ribosomes from RER, then SER basically forms. The ER membrane is thinner by 50 angstrom than that of the plasma membrane. Means ER is 50 angstrom, then plasma membrane is 80 to 100 angstrom thick. Now we will see the physical structure of ER. The physical structure, as we know, comprises a 3D dimensional network of intracellular. It is formed of three types of elements. One is cisternae, second is tubules and vesicles.